All right. Well, thank you all for attending, especially at this time of year, so close to the holidays. We, the small team working on helping Blane document his methods, wanted to have this meeting before he retires. And if I count correctly, I think the number of days you have left, Blane, can be counted on two hands, maybe one. I don't know your schedule exactly. So let's get started. Today we'll be presenting on the NMFS method of estimating CRS adult conversion rates, and this includes documentation of the method, continued access via DART, data visualizations, and some thoughts on potential next steps. And I'd like to acknowledge the authors, first and foremost Blane, coming out of NMFS West Coast Regional Office in Portland, Russell Scranton, BPA, myself, Matt Carter, Susannah Iltis from CBR DART, Kate Self from NPCC, and Brian Maschhoff from Salmonetics. So a little bit of background before I pass it off to, and give an outline and pass it off to Blane. A little bit of background of how we got here. So as you all know, conversion rates, those are an important salmon metric in the region, and Blane has been reporting these for BIOP reporting. The method was first developed by Charlie Paulson in the early 2000s. The method was then passed on to NMFS, specifically Blane, and he took over in 2008. And recently, because of Blane's upcoming retirement, UW CBR DART was asked to help maintain the method and continue providing access to these estimates via the DART website. So a quick outline of what we'll be presenting on today. Blane will start with an overview of his methods and what he's been working on in terms of knowledge transfer, essentially, before retirement. This includes written documentation of his methods and files for historical data sets and estimates. And then we will then share how and where the written documentation and data files can be accessed for the NMFS method that covers the adult return years 2009 through 2023, and then the DART applied NMFS method from 2009 to present. In the third section of our presentation, Kate Self will share the NPCC strategy performance indicators, specifically for conversion rates. I'll give an overview of what currently exists for DART conversion rate tools. Brian will share what currently exists for one fish, two fish conversion rate tools, and we'll mention a few thoughts on what can be improved or better integrated. And in the last section, we'll delve a little more on potential improvements related to data and analysis, as well as potential ways of hearing feedback and regional collaboration. And with that, I will pass it on to Blane, who will cover the NMFS method for conversion rates.
Okay, while I'm thinking about it, I want to express my gratitude to Susannah, who's here, for all the support she's given me through the years and helped me out. And then also, they aren't here, but the PTAGIS staff was also very helpful and helped me out when I got in tight spots and couldn't figure things out. Okay, this is part of the Adaptive Management Program. It was meant to monitor adult survival and especially detect changes in adult upstream survival and use, if we're meeting the BIOP adult upstream survival standards. It was also provided to, I think it was BPA and the Corps for their annual reports, and then provided, well, at first it was kind of the first one that we had beyond just window counts. And then from then on, it was the comparison to the last BIOP as we went through time. Next one, please. So we have a unique situation here in the Columbia Basin. I always tout it heavily whenever I give talks elsewhere of a series of highly efficient detectors, especially for adults. And then beyond that, a whole series of other detectors which everyone shares their data, puts it in a common PTAGIS database. And this is very unique, and people usually look at me with envy from the East Coast and other places like that. Next slide. And there's some of the sites. You're all probably pretty familiar with all these sites. Go ahead, the next one, please. So this came about this whole thing is kind of a value added project. Charlie Paulson realized we have large numbers of known origin pit tag adults that return at Bonneville Dam every year. As far as I know, none of these were tagged with a specific intent of estimating adult upstream survival, though some of them were definitely, excuse me, some of them were definitely tagged for purposes of, estimating SARs, which adult survival is a component of that. And so we do have a fairly generous number, so we can have a good sample size every year. Snake River sockeye are a bit of an exception to that rule, but we have a lot of data to work with. Next slide. So what we've used historically is just a simple binomial method. The number of fish at the upstream site over the number of fish at the downstream site, and that gives you the conversion estimate. Actually, this estimate includes the detection probability. It's rolled in when you do it this way. But since the ladder detectors tend to be very efficient, 97 to 100%, it makes a pretty good workable estimate of survival, and it's also very transparent. You don't have to have a lot of specialist knowledge to understand what went into it, and it gets better trust in the management world, is what we've found. So the key assumption in any of these analyses is you only can use fish that are expected to arrive at the upstream detector. So we avoid issues with turnoff. That's the advantage to using known origin fish. All the fish from the Snake River should try to go back to the Snake River. Okay. The data was downloaded from the PTAGIS database, developed some basic queries, which have changed over time, mostly reflecting my changes in workflow and hopefully efficiency. And then I used custom access database and queries to filter the data and assemble the groups for analysis by ESU, dropout, jacks and mini jacks, and those sorts of things. And then the reporting was by an Excel spreadsheet, which is the same one since Charlie handed over to me in 2008. And it exists in the archives, somewhat cleaned up. So this, if you go into the archives and look at it, this is presented as a historical document, warts and all. It had some additional extra analyses that I'd thrown in that were kind of my own, but that's all been removed. So a few other things like the harvest estimates, those are snapshots based on the year that they're published, because the harvest people go back and update and correct their estimates. So it's a historical document. That's what was used in the BIOPS. One of the advantages to this data is if you don't like the way I did it, all the data still exists. So you can go back and recalculate it, as far back as you want to go. Next slide, please. Okay. So each year I'd assemble my group at Bonneville, which is the start of the analysis, by doing the queries from PTAGIS. And then there's some regular processes you have to go through. You have to eliminate duplicates. Bonneville is a real problem because the 4 detectors at the 2 ladders, and it's kind of plumber's nightmare of the way the ladders are set up, you end up with duplicate detection. So I had to come up with some methods to choose one detection out of all those. And then there's also fallback and reascension issues that are possibly happening. So that can cause duplicates. This happens at the other dams, mainly in terms of the potential for fallback and reascension. Next slide. Okay, so once you get the data, filter it by age. I would sometimes get rid of mini jacks before I downloaded it. It's possible to do that in PTAGIS, but I generally keep jacks because there's some interesting comparisons that can be done, though they aren't part of the reporting standard. So I created a ocean age, and you see from there, that's the first year from the migration year, which are part of the PTAGIS metadata. Assign the fishes to the ESUs based on a custom access crosstalk table, which takes the release point and connects it directly to an ESU. This was something that it took a long time to put together, but it was definitely worth the effort. Identify juvenile migration history. That is our main concern, and because it's part of the BIOP requirements, was if they migrated in river or transported, because that was part of the ongoing adaptive management measures to see if transport measures were being effective. And there was a lot of questions about straying by transported fish, which they do tend to have slightly lower conversion overall, though in some years they actually have better conversion than river fish. So it does appear to be an issue, but perhaps not as big a one as a lot of people seem to think. But the analysis was designed to allow us to examine that. And I did that originally by, I don't even know if I can dignify it by calling it an algorithm, but my own procedure where you go through and you first take the transport window, and assume that all the fish that went in the bypass were transported. And then from there, you keep applying filters to it to reduce it. Try to get rid of all the ones that were returned to river, which is relatively easy. You got to sort through by last antenna and then all the way down, try to account for the dump days. You can go into the FPC data. A lot of times it was a very involved process. And fortunately, it turned out that DART was doing it, and they have the skill and ability to mechanize the process. So from then on, I called up Susannah and asked her for it, and she provided it to me. One thing you should remember in all these are all estimates. If a fish was transported or not, there isn't any direct evidence. I know the Corps put a detector, I believe, on the loading station for the lower granite barge, but I've never looked at what kind of data that produced. Next one, please. So it can be very important to what ages you include, especially now that for a particular year class, half of them will be returning as jacks or mini jacks, especially for hatchery stocks. So the tendency you see is, especially in the lower river, which is the x-axis or the dams as you go upstream, jacks tend to survive at a higher rate than adults in the lower river. And part of that is probably due to they aren't quite as susceptible to harvest because of gillnet selectivity. I've never proven that or looked at it too close. And then mini jacks, they just don't do as well. So that can really bias your results if you keep them in there. And they're probably more subject to predation. They don't have the energy reserves to make the trip. They can spawn. I've seen them spawn successfully. But in case you don't know, mini jacks are fish that spend less than 1 year at the ocean. In fact, from some of the data I saw in Ptagus, they leave Bonneville, spend a couple months downstream, I don't know, go out and taste salt water and turn around and come back. So it's a very short period. Next slide, please. Okay, so once I have my Bonneville tag list, that's my start list, and download that to the Access database, and then I use queries to assemble. Well, I also do a query for all the observations at adult ladders in the CRS. So I use that Bonneville list to make that query. And so then I have a whole set of all the observations, and then I use further queries to assign the fish, each fish and each observation. So I end up with something that looks like a CGS input file, more or less, if you've ever seen one of those. So I got the pit tag number, if the fish is transported in river, and then like a 1 or a 0. In the end, I have a 1 or a 0 if it was observed at Bonneville, the Dalles, all the way up, dam by dam. And then from there, the numbers are tabulated. So fish are individually tracked through the system rather than just going number to number to number. Next slide. Sample size, we came up with a minimum size estimate, a minimum sample estimate of 40 tags at Bonneville. As you can see there, if you have more, the uncertainty tends to blow up on you. 40 tags was pretty easy standard to meet for everything but Snake River sockeye. And for them, we ended up having to use Upper Columbia sockeye as surrogates, especially in the earlier days before 2015, before the Snake River sockeye program really took off. And there were a lot of pit tag Upper Columbia sockeye in the river because of the PUD projects on the Upper Columbia. We're conducting some studies. We had to do it again recently because of the issues with one of the hatcheries. We had some real low returns, but they seem to be coming back from that again. Next slide. So our reporting reaches, Bonneville Lower Granite, Bonneville McNary, McNary Lower Granite. So these, when this first started, these were really all that was possible because of the number of detectors. So all the fish that would pass through the whole CRS were reported for those three reaches, and then Upper Columbia fish and Middle Columbia steelhead, an estimate was only made for Bonneville McNary, because after that they leave the CRS. We might have done analyses to track them further upstream just for other questions and to see what's happening, but that was not reported officially or included in any of the BIOPS. Next slide. Here's a reporting spreadsheet, which is probably impossible to see. Basically, it walks you through. It has the numbers at Bonneville, McNary, and Lower Granite, calculates a raw survival, applies correction for harvest in zone 6, which is Bonneville, McNary, straying estimate, which came from a study, 2004 study by Kiefer, and that's constant and the same for both in-river and transported, which is the difference between the two tables. The pink or salmon color are transported, the blue are in-river, and then any Snake River harvest, if that occurred, was also accounted for. And those corrections are made, and then it's applied to give you a corrected estimate. Let's see, what else do we have? Okay. And I guess that's basically what I already said. Harvest corrections came from the TAC reports, and that was a little bit of an estimate because their reporting groups and periods don't precisely match up with the ESUs that we're interested in. So it's a little bit of a struggle sometimes. So those are also something that's, once again, an estimate. And TAC does go back as they get more and more data. Some things like steelhead catch cards, they don't get them till quite a bit later. And so they can't incorporate them until later years. And then there's the citation for the straying study. That's another thing that could be improved. There have been further studies. Maybe you can come up with something to apply different rates to transported in-river fish. But this was held standard. Part of this too, when it's in the management frame and we're measuring performance, is you need to avoid being accused of moving the goalposts. Sorry, we got a family thing going on. To avoid moving goalposts, and so you keep your standard of comparison constant. Next one. Okay. Oh yeah, some other twists that come into play. Snake River steelhead, a significant portion of them will overwinter in the lower river. So we came up with, and then the fact that the distribution of summer steelhead crosses a calendar year. Nobody told them about the calendar apparently. So your typical summer steelhead will enter the river in late summer and then spawn the following spring. And so we established a steelhead year of June 1st to May 30th. And that doesn't quite match up with the one that TAC did, but that seemed to be from the data where the natural break was. And also the Snake River steelhead populations include some really late spawning groups. So they could still, even if they, cross Bonneville May 30th, they could probably still make it up in time to spawn. And then noted the problems with Snake River sockeye pit tags. Next one, please. And so, yeah, the estimate is an average for the entire migration period. Fish were not the fish for analysis once they were classified as particular ESU were not constrained by dates. Anytime during that calendar year that they showed up and were detected, they were counted. Some of the questions are how representative are these tags? Because as I said, we're doing a kind of a value added follow on. I did make an effort to be careful of any issues or treatments that might have been done that would bias these fish. However, the fact that they spent 2 to 4 years in the ocean and then have returned probably weeded out a lot of those effects, I assumed. The issue of surrogates and enough tags, we actually use this similar method for a lot of in-season management questions, but it's always the question of, do you have enough tags? Because you get down below that 40%, you get some pretty unreliable results. And then once again, we don't have any control over the distribution of tags. From comparing the tag detection rates to the return curves, it looks pretty close, but that's another potential issue. Some small populations could be getting short shrift, especially if, a lot of them, they don't have anywhere near enough pit tags returning to be independently evaluated. Next slide. Other methodologies and options, Cormac Jolly Seaver, DART conversion tool, which I believe also uses binomial method, but that's a good tool. Pit Tag Pro and Surf software are made available by DART and UW CBR. And if you want to use CJS analysis, it's designed for that. The only problem I found was apparently the programmers never imagined that you'd have 100% detection efficiency, and it kind of loses its mind when it gets those values, but you just got to know that's what's happening and just go ahead and do the binomial method. Next one.
Thanks, Blaine. Yeah, thank you very much. So moving on to the documentation and access to Blaine's method, data and estimates. In the next several slides, we'll be sharing some screenshots and website URLs. And this will include archive files for the NMFS method and documentation and data files for the DART applied NMFS method. So the easiest way to access the files is currently through the web page for the DART applied NMFS method. This is a new web page on DART. Here is a simple query you can run to access the data and estimates files. And there's also, you can see at the bottom of the screenshot, there's a link to the GitHub repo for documentation. So when you get to the GitHub repo, it'll look like this. And clicking on the methods folder, that will bring you to documentation and archived files for the NMFS method, including Charlie Paulson's archived files. As well, there's documentation and data files for the DART applied NMFS method. So when you click in and you go into the folder system for the NMFS method, this is what you'll see. It includes Blaine's written documentation, that's about a dozen pages, a copy of a presentation he gave earlier this year, which is very similar to what he just presented, some Excel files of the data he used, and we'll include some more years in that folder shortly, and Excel file for the conversion rates estimates. What you don't see in the screenshot, there's also a fourth folder, and that's where you can access Charlie Paulson's archived files. In another folder system that's specific to the DART applied NMFS method, you'll see this. There's a folder for the DART, for all the data files that go in and the conversion rate estimates, and also a written documentation for how we translated Blaine's method for an automated process within DART. So a little bit more about that. The documentation is a couple of pages. Some small differences include incorporating the DART ESU filter, which we already have in-house, and apply it to a whole bunch of other DART tools. So we thought it would be easiest to apply it to this method as well. Blane mentioned that he already uses Susannah's DART transportation filter, and so we continue to use that here. Drawing directly from TAC reports, harvest estimates from specific reports, and we try to be as transparent as possible in citing which year's reports and which tables. And as Blane mentioned, as those get updated, we plan to update those estimates and incorporate them into these estimates as well. So for that reason, as data and estimates change through the years, there might be some slight differences in the pit data and the harvest rates that are applied in this method compared to what Blane applied in each year that the adults returned historically. So we'll try to be as good as possible in terms of versioning those methods and documenting those changes. Onto the third section now, access to the conversion rate estimates, data visualizations. We'll go over some of those now. Already planned and in development presently, there are the NPCC SPIs, and Kate will present on this next, and then we'll go over some DART and one fish, two fish tools. Kate?
Okay, thank you. This is very brief. I just wanted to remind this group and give people who haven't checked it out before a flavor of what's available through our strategy performance indicators on our program tracker. So this is on our main website. A group, my colleagues and I, and working with regional managers have been trying to, well, the program tracker has always been updated, but really working to make it more accessible and more intuitive ahead of this upcoming amendment process for the Fish and Wildlife Program that will launch mid-January. And the strategy performance indicators draw directly from our program goals and objectives and measures. And so this data that we're talking about today is part of that, because our Fish and Wildlife Program is more expansive and comprehensive than BIOPS themselves, but BIOPS are folded into our program regularly whenever the program is amended. So this data directly is part of our program as well. And this is just some screenshots of what the program tracker for this data will look like if you navigate there. We just offer kind of, this is a simplified version, I would say, compared to some of the other resources that are out there. So people who are looking at a glance to get an idea about trend or download the data, the Excel tables are also available for download, as well as exporting these charts if you need them for any of your presentations or just for reference, and covering many of the available estimates that Blane and now others will be providing into the future. Yeah, just a plug for our program tracker that has many other resources as well, but this is an easy way just to kind of check in on conversion rates and update it on an annual basis. So we really I appreciated being included in this group over the last 3 or 4 months, just because my previous life was harvest, and now I'm working on hydro system stuff at the council and also on this program tracker. So it fit really nicely in keeping us up to date with the future changes. So congratulations to Blane and everybody else that's been working on this. And feel free to reach out anytime if you have general questions about the program tracker or these conversion rate tracker specifics. So I hope that's enough information, but let me know if you have any questions.
Thank you very much, Kate. For the DART conversion rate tool, here's a screenshot of the query interface. As you can see, users can see the data attribution for the pit data. They can select reach, species, run, and rear type of interest. They can choose to exclude or include upstream detections. As well, they can choose to exclude or include mini jacks, release sites below the project of interest, and transported fish. Therefore, users can specify their customized selections before hitting the submit button to run the query. Also, a few things to mention: there is no minimum number of fish in the query, and it's only at a high level of ESU or DPS. So there are some potential areas of refinements that we can work on. Once a query is run, a user can see a graphical output like this. It includes, in orange, the annual pooled conversion rate estimates with the 95% confidence interval. And for comparison, there's the dotted orange line for all the historical estimate there. Down below in gray and associated with the right y-axis is the annual Bonneville weighted average arrival date, and the transparent color there is the arrival date and the first to last dates of arrival in each year. Users can also access more detailed information via tables, and that includes data on release site, RKMs, date, sample sizes, and detailed detection details. The DART conversion rate tool also includes comparisons by species, run, rear type, as well reach comparisons and transport comparisons. So there are many options for users to access the data, the visualizations, and comparisons. And I'll now pass it on to Brian, who draws from these data for additional tools that he develops.
Hi there. So what my work has been focusing on developing concepts for looking at the data in different ways and making use of DART data. So the first one basically takes data straight from DART, and as the main goal was to try to look at all comparisons between all dams across all years rather than look at 1 year at a time and figure out ways to do that. And so it's sort of a zoom out type of concept where we're just kind of looking at the bigger picture, what's going on. The second one is going the opposite way, is looking within a year, because looking in the first case, you identify a lot of differences between different individual reaches, what's going on, that kind of beg for, okay, the goal is to try to identify reasons that we have variable conversion rates across reaches. And to do that, in looking at the data, you identify that it's not a uniform, things are up this year, down this year. There are specific things that are going on that we're trying to identify, whether it's flow conditions or spill conditions that might be causing, or at least somewhat responsible for, changes in conversion rate. So that will require going beyond what is easily available for the DART system. And so I would download data from Batagas for this. So move on to the next slide, please. So this is a couple of screenshots from the first of these two tools. And what you're looking at in the lower left is really looking at changes relative to a mean. In this case, it's a mean from like a 10-year, or what is that, 1, 2, 3, 4, 5, from 2015 to 2023. And so there's a row for each of the upstream going from toward Bonneville toward Lower Granite for each of the individual reaches between dams, as well as a composite for the full reach, Bonneville to Lower Granite. This is for Chinook. And basically looking at changes relative to the mean for each of these reaches, you can identify differences across time that are more easily apparent just by looking at the data, as well as looking at the individual numbers and confidence intervals. And looking at the upper right, it's all like a 500, or 500-mile view of what's going on. Okay, how can we come up with some way of looking at the things that we're observing across reaches, identify where a lot of the fish are being, most of the fish are being lost. And so there's various ways of kind of visualizing the raw numbers on a map display. One caveat with this particular tool is that it's somewhat rough around the edges. It's more been developed as a concept, so it's not bulletproof, but it does provide a lot of insight in what might be going on across all the reaches in a combined view. Next slide, please. Next slide.
It should be updating. It might take some time to update
Okay,
on
there we
the
go.
slide.
So what I was interested is looking at within 1 year, how does the apparent conversion rate change? Um, and so there's and also how does that compare with what might be going on in terms of the environment, whether it's spill or temperature or flow. And so this tool has a lot of little widgets that help kind of look at that for a given for a given year and given species. So you can select a variety of groups on the left there, whether it's ESUs or release zone species. Um, and there's also in this tool an annual summary, which kind of mirrors what the DART tool does for a given species in a given year and given reach. But also you can select an individual year down on the very lower left, and then you get a variety of tools to explore that. So at the top you see sort of an example of how the rate kind of changes as the year goes on for Chinook. And then you can look at a variety of different environmental covariates to see if there's something that jumps out. For the most part, it doesn't. It's kind of a little bit still kind of murky. But the real key is to be able to look at the individual fish, which is kind of in the upper right there. It's a little bit hard to see, but it's basically a dot for every fish that has been detected at the lower dam site. The red dots indicate that the fish never made it, so those are the ones that did not converge. The other dots are related to or basically graphed where the travel time that it took between detection at the lower dam and the upper dam. And so with a scrubbing tool, you can select certain groups of fish to identify, look at various statistics, including conversion rate for that particular group of fish, or the mean travel time. And the goal there, or the utility of this, is to really deep dive into various parts of the year to see if there are certain conditions or certain things going on at certain periods which give rise to the changes in the annual conversion rate. So the main takeaway is that we look at annual conversion rate as a metric, but there's a lot going on under the hood that really determines what that is. And it's if we are going to go beyond just using it as a raw number as a metric, but actually try to understand what are the causative factors for lower conversion rate for a particular year for a given reach, requires a little bit more detailed data and different ways of looking at the problem. So in summary, both of these tools are really meant for trying to start the process of asking questions about the data. You can look at different groups of fish and apply all kinds of filters, but at the end, the goal is to try to help identify strategies for using the information that's available from DART and from Batagis to try to better understand what's going on with conversion rate, travel time, and all these other indicators of upstream performance.
Thank you very much, Brian. I think this leads well to the next and last section as we suggest some ideas and possible next steps related to the data and the methods and regional collaboration. And we've been focusing on documentation of methods and recreating Blane's method for automation. And we're starting to think about what possible next steps there can be. So for data, some updates on stray rates could be incorporated, drawing from most recent published studies and reports. Conversion rates estimates can be broken down further by population, rear type and passage type for sockeye possibly, smaller reaches, and so forth. So breaking down the data further. and including covariate data that could be compiled as well, compiled in a way that matches the fish data and some considerations for underlying mechanisms, like Brian just mentioned. For analysis, a CJS model could be applied, perhaps also in a Bayesian context. Covariates could be included in this model. As models of harvest rates improve, those could be included too. And there's potential for refining models of stray rates for better estimates as well. So those are some ideas. In terms of data visualization tools, there are many that exist already, as you can see, from DART and from One Fish, Two Fish. And so integration and consolidation of many options that are available could be possible, and that would depend on feedback and needs from action agencies and regional co-managers. And on that note, FPOM could be a place for discussions because it sounds like there's interest already. We've been focusing on documentation up until this point, and potentially we can now learn more about what can be improved from current methods. We're open to having additional meetings or forums for discussion, and we'll try our best to provide that information and tools that are needed. Some considerations, if these next steps occur, would be identifying who would coordinate these meetings in the process, and we could learn more about regional needs related to conversion rates and what's useful in terms of integrated online tools for more centrally located resources. So again, our aim is to assist action agencies, state and tribal co-managers as best as we can with the time and capacity that we have, and it'd be good to hear more about what's of interest and what's needed. Russell, please feel free to chime in if there's anything else that I missed.
Oh, and the last piece is, again, just maintaining, again, this is the main purpose is maintaining this data set for continued use and acquisition of the conversion rates as BIOP discussions continue. So I think that will be the last step is just with the AAA's NOAA, just continuing to identify points of contact to discuss this with DART moving forward.
Thanks.
Thank you.
Thank you, Russell. And with that, we'd like to acknowledge Patajas, PSMFC for the pit tag data, as well all the folks who worked so hard to tag the millions of fish in the Columbia River Basin. I'd like to thank Charlie Paulson for developing the original methods, folks at the Northwest Fisheries Science Center for assisting Blane and others, Melanie Chen at CBR for assisting with the documentation for the DART applied NMFS method. And we'd like to thank the Action Agency, the Corps, BPA, as well as NOAA for your interest and input. Any feedback you provide would be very much appreciated. And we thank BPA for providing funding to conduct DART and One Fish, Two Fish work. With that, we'd be happy to take any questions that you have.